we're going to shift gears here and talk a little bit about some regulatory concerns and issues and challenges. And to set that up, I want to offer up polling question number 2. Let's start by asking this question of our audience. Is it your impression that adopting intensification in your upstream processes will ultimately ease your regulatory requirements, immediately create regulatory challenges, or result in little to no change to regulatory requirements? Go ahead and consider those questions, make your selection now, and we'll come back and revisit that, the results of that poll shortly. In the meantime, I want to pose a similar question to Parrish and Susan. New technology and new processes don't go unnoticed by regulatory agencies. So what are the regulatory and quality considerations associated with upstream process intensification initiatives? Susan, let's start with you. What regulatory, top of mind, in your face sort of regulatory issues did you have to address right off the bat?
It's hard to say because we're at early stages, but I can tell you that what we saw in differences between product quality from a fed batch process versus perfusion was that we had a better product quality in perfusion than we did in fed batch. And so now is that a comparable molecule to the original one? And so when we wanted to transition from fed batch to perfusion, we had to characterize the molecule, look at the glycan pattern, look at the peptide map, look at all the characterization parameters in order to show that we hadn't really, that the molecule after purification was essentially the same. But there are things that are different that will affect the molecule in terms of your main peaks and your acid and base peaks, et cetera, that are just fundamentally because of the perfusion process. So I think if you start out in perfusion, if you develop a perfusion process and your product quality is your reference standard, then I don't see any major hurdles from a regulatory perspective. But again, I'm not going for months and months, and I'm not trying to extend my manufacturing process to create a batch that's not a well-defined batch. My batches are well-defined, which I think is one of the biggest kind of gray areas of how do you define a batch. I have a very clear batch definition for my molecules, so don't have that regulatory hurdle, which I think is probably a bit more risky and a little bit more obscure.
Mm-hmm. Parrish, I'd like you to weigh in, and maybe more specifically, either one of you, if you want to address the question, how does implementing N1 perfusion impact regulatory paperwork in an existing fed batch process?
Well, it could require an amendment to your IND or to your commercial process if you're implementing after the fact. But in fact, the regulatory bodies, agencies have been very positive on continuous manufacturing. They promote it as a way to increase product quality and potentially reduce production costs, which increases access for patients to the drug. Mm-hmm. So we have not run into any regulatory hurdles with the regulatory agencies in implementing perfusion processes so far.
Okay. Let's say I have a current facility with fed batch equipment and I want to implement perfusion. What kind of validation must I perform?
Well, like any change to the process, you need to do an IQ, OQ validation of the equipment. And then once that's performed, and if it's proved adequate, and met requirements, then the next step is to do a performance qualification by running a real culture through the process and validating the performance of the system to achieve the cell density and product quality goals that you had upon embarking on the project.
Okay, let's take a look at the results from our poll and see if they jibe with where this regulatory conversation is going. So 8.8% anticipate that adopting PI will ultimately ease the regulatory requirements. 35% anticipate that it would immediately create regulatory challenges, and 56% anticipate no or little change to regulatory requirements. Parrish, what's your response there? What's your take on those results?
Well, I agree with the 56% no regulatory hurdles. As I mentioned, the regulatory agencies have been very vocal about supporting continuous manufacturing from the perspective of reducing costs and also increasing product quality and consistency.
Mm-hmm. Okay. Can high cell density banking remain where there's an already established process for it? And what are the regulatory implications for that?
Well, if you're changing the way you're banking your cells, you obviously need to validate the stability of those cells using the new method. So if you're increasing the cell density in the cell bank so that you can save time in inoculum preparation, you need to validate the stability of those cells under those high cell density conditions. And that would mean confirming their productivity over the extended life of the cells in manufacturing and long-term stability over at least 3 years in liquid nitrogen.
Susan, tell us, I understand you guys, Sonnet has not considered high cell density banking. Tell us about the banking environment there at Sonnet.
Yeah, so actually we did implement I don't know if it's the highest cell banking densities, but we did more than triple the cell densities that are typical of cell banks. Okay. In fact, we might have even gone higher than that, maybe 5-fold higher. And we actually find that we can knock a few days off manufacturing. So I think we've been able to get in the N-1, actually in perfusion, the time zero escalation to really good productivity levels and maintain those productivity levels for a few extra days. For example, instead of hitting peak cell density at day 8, we're hitting it at day 6. But we've knocked off the front end of the process so we get to those high cell densities fairly quickly. And it's produced good quality drug very consistently across the manufacturing duration. So it's benefited us by knocking off about 3 to 5 days. It depends on I've got 3 molecules. It's 1 day for one of them, but it's 5 days for another one of them. Off the duration of the manufacturing process, which can be a lot. It adds up.
Sure. Yeah, no doubt.
So, so we like the high cell density cell bank approach.
Yeah. Okay. Again, in the context of interactions with the agency, what's a major change versus a minor change when a molecule is already in certain phases, and what kind of confirmation runs or studies must be performed?
I guess I would say that if you change product quality and it affects potency by improving a potency, you need to do some work to show that in fact it is potency and not something else that's been improved, that you actually have a product quality improvement and not a difference in the product. I guess the other thing I would say is I forgot what I was going to say.
We can turn to Parrish.
Yeah, turn to Parrish. It'll come back to me.
yeah, for sure. Yep. Major change versus minor change. How do you qualify those two things, and what kind of confirmation runs and studies need to be performed, Parrish?
Well, if you were doing production in fed batch through phase 1 and phase 2 clinical trials, and then you decide to change to perfusion for phase 3, that's a major change to the process. And validating that would be required. You would have to produce product using the perfusion process. You would have to characterize the product quality. And if the product quality matches the original product, then you're good to go and can submit a comparability protocol to the agency for review and approval. If the product quality does not match the original product, then you've got a real challenge now to evaluate what the impact is on safety and also potentially bioactivity or efficacy, as well as PK/PD in humans. So it depends on the result of what you see as you embark on this, with regard to what's the regulatory impact that you're going to experience.